Hi everyone, now we're on part B of chapter 21, the immune system. So we're going to finish up with part B, and this of course is talking about primarily about antibodies. So what's an antibody? Antibody is called an immune globulin. A globulin is kind of a globular shaped protein, so it's a protein that travels in the blood, and it's called a gamma globulin because it's shaped like a Y. The Greek letter gamma is shaped like this, and that's what they're shaped like. So here's the Greek alphabet. I thought I would pull that up for you. Alpha, beta, gamma, it's a Y shape. These immune globulins, called gamma globulin, are secreted by plasma B cells. Remember, the plasma B cell is the effector cell. That is the type of B cell that actually produces and secretes the antibody or gamma globulin. These antibodies bind specifically with that antigen, that foreign marker that's recognized by the body, that it is something that has to be destroyed. There's actually 5 different immune globulin or gamma globulin classes. IgM is actually the first released. It fixes and activates complement. Then there's IgA. IgA is found in mucus and other secretions. IgD is found on a B cell surface, and it's an antigen receptor. And IgG, this actually forms about 75 to 85% of antibodies. It's active during the secondary and late primary responses, and it does cross the placenta. So this is one that during breastfeeding the mother will pass to her infant to protect it against certain pathogens that she herself has developed immunity to. Then there's IgE. IgE is released during allergies and parasitic infections. It actually binds mast cells. Mast cells produce this chemical called histamine that's involved with inflammation and allergic responses. And it also basophils. Basophil is a type of white blood cell that will greatly increase in the bloodstream during an allergic response. Basic antibody structure is either a T or a Y-shaped antibody monomer, meaning the major part of it. It actually has 4 polypeptide chains that are linked by disulfide bonds, and there's 2 identical heavy chains, H chains. They hinge in the middle, and then 2 identical light or L chains. At the end of each arm, like at the tips of the Y, the 2 parts of the Y that stick up, these are variable regions. So we call these the arms, at the tips of the arms. These variable regions form 2 identical antigen binding sites. Oops. So the antibody structure has a constant regions of the stem, and this is going to determine whether it's IgM, IgA, IgD, IgG, or IgE. And you don't need to know all the details of which type of immune globulin does what, but there is a table that summarizes them toward the end. Understand that there are types of basic structures to antibodies. There's the 5 different classes. And this is showing you the basic shape of an antibody, which I do want you to remember. These are involved in humoral immunity, meaning the antibody mediated. You have this, right here you've got your constant region. You've got the stem region with the hinge. Here's the heavy chain and the light chain on each side forming the Y. And so this is your basic shape. It's even showing you the disulfide bonds there, the basic Y shape that leads to the name gamma globulin. This is showing table 21.4. There's actually 2 parts to this, 2 slides. Immunoglobulin classes. This would be your IgM, which it looks like a big star shape, and it talks about it's actually very early in the primary response. Then you have IgA here, and here's IgD, IgG, and IgE. So antibody targets and functions. Antibodies will inactivate and tag an antigen. It forms the antigen-antibody complex. The defense mechanisms include steps, neutralization and agglutination, precipitation, and complement fixation. And here is showing these mechanisms of the antibody action just mentioned. The antigen comes into the body, and the antibody, these little Y-shaped molecules here, bind to it, forms an antigen-antibody complex. It inactivates the antigen through neutralization. In other words, it masks the dangerous parts of the bacterial exotoxins or viruses. It agglutinates it, so it's going to cause these molecules to act, any kind of cell-bound antigen, it pulls them all together and forms it agglutinates it, pulls them together and forms clumps. Then it precipitates. Okay, so this is actually, and this would be for soluble antigens that are in the bloodstream. It'll actually come out of the solution and, basically makes it so that it can't infect the body. This is actually going to lead to an enhanced phagocytosis. And so that way the white blood cells will come over and actually just munch away and neutralize, get rid of those antigens. And you can see these phagocytes identifying and destroying this complex. This antigen-antibody complex will fix and activate complements. And this complement is actually going to enhance inflammation, okay, and allows the histamine to be released. It encourages chemotaxis, so the macrophages will go over to neutralize the pathogen, and it will also lead to cell lysis. So this would be like a bacterium right here, and this is showing lysis, that it will actually be destroyed and neutralized. There's actually, in medical research, there's something known as a monoclonal antibody. They're used as clinical and research tools. They actually have produced what's called a hybridoma. It's the fusion of a tumor cell and a B cell. This will proliferate or multiply indefinitely to produce a single type of antibody. T cells actually are a defense against intracellular means within the cell, against intracellular antigens. Some will kill the cells directly. Others release chemicals that will regulate the immune response. There's actually two major populations of T cells. CD4 cells. These usually become helper T cells. Some become suppressor or regulatory T cells, or they can become memory cells. And then there's CD8 cells. These will mostly become cytotoxic or killer T cells. They will destroy the cells that are harboring these foreign antigens. Some of these may also become memory T cells. So if these are two different populations, eventually these give rise to those four types of T cells that I mentioned before. You have helper T cells, regulatory or suppressor T cells, and that's actually, these will form once a CD4 cell encounters some type of an antigen, and then this population of CD8 cells becomes the cytotoxic T cell. So actually you've got one helper T cell, two regulatory or suppressor T cell, three would be the memory T cell, you can also see those here, and then four would be the cytotoxic T cell. These are summarized later as well. So CD4, CD8 cells are actually naive cells. This is before they've encountered an antigen. And here is the actual classification by function. Once they become specialized, once they are no longer naive, they've encountered an antigen of some type, they will then become cytotoxic or killer T cells, helper T cells, regulatory or suppressor T cells, or memory T cells. So we have all types of these four T cells that will be engaged in fighting the infection. And this is a figure that gives a summary. You have an immature lymphocyte. This would be for a T cell. It actually will mature into a CD4 or a CD8 cell. And they're showing how CD4 cells will be activated, and these can give rise to memory cells. They can become helper or regulatory cells. And then they're showing the CD8 cell. When it's activated, it will become, it will specialize to become primarily cytotoxic T cells. Some of these will also become memory cells. And this is a further discussion of the major histocompatibility proteins and antigen presentation. T cells will respond to processed fragments of displayed antigens. Remember those antigenic determinants. The antigen presentation is vital in order to activate naive T cells and effector T cells functioning. There's something called a cytokine. A cytokine is the immune system's chemical messenger. The cytokine mediates the immune system's responses. It actually leads to the production of interferons and interleukins. And actually, this is now going to go over the roles of helper T cells. Helper T cells play a central role in the immune response. These actually activate both the humoral, the antibody-mediated, and the cellular or cell-mediated arms. Once primed by the antigen-presenting cell, presentation, this is what is going to activate the helper T cell. What this does, it will increase the T and B cell proliferation and help activate them. It will recruit other immune cells. If you have no helper T cell, you get no immune response. This is actually why with HIV, human immunodeficiency virus, it actually infects CD4 cells. So therefore, there's no helper T cells. So you not only reduce or eliminate cell-mediated immunity, but you also eliminate the humorally or antibody-mediated immunity. And this is why individuals that are infected with HIV can die of infections or types of cancers that the body just is not able to fight off. They have lack of an immune system. So this is showing helper T cells helping in humoral immunity. The helper T cell binds with the self-non-self complex of a B cell that has encountered its antigen and is displaying it on the MHC2 on its surface. So this is showing you the helper T cell right here that's going to release interleukins to signal the complete B cell activation. So there's a lot of detail here. Just keep in mind that the helper T cell is going to bind with any kind of non-self complex that a B cell's encountered and will actually display it to allow antibody to be produced, and will also release interleukins to stimulate signals to complete the B cell activation. Now here's the helper T cell helping in cellular immunity. So the helper T cell binds a dendritic cell. The helper T cell stimulates the dendritic cell to express co-stimulatory molecules, and now the dendritic cell can activate the CD8 cell. And then the CD8 cell, with the help of interleukin 2 secreted by the helper T cell, will become a cytotoxic or a killer T cell. Cytotoxic T cells, it is that big T with a little C subscript. These directly attack and kill other cells. Activated cytotoxic T cells circulate in the blood, the lymph, and lymphoid organs. They search for body cells displaying familiar antigens. And so anyway, if they're self antigens, they will leave them alone. If they are non-self, and they will recognize them as foreign. The roles of cytotoxic T cells would be that they target a cell that is displaying signals of being infected with a virus. Cells that have intracellular bacteria or parasites. Remember, intracellular means within that cell. So a cell can have bacteria or a parasite inside it. Also a cancer cell, which is an abnormal cell, or a foreign cell, like with some type of a tissue transplantation. The cytotoxic T cell targets all of these types of cells. The cytotoxic T cell destroys all infected or abnormal cells. And so this is actually showing you a cytotoxic T cell that's attacking an infected cell and a cancerous cells. So it identifies the foreign antigen on the MHC1 proteins, and it binds to that target cell. And then the cytotoxic T cells releases perforin. Perforin is a substance that perforates, literally pokes holes in the cell, and then granzyme molecules. And this will actually break down and destroy that cell. The perforin molecules insert into the target cell membrane, polymerize, and form transmembrane pores. They actually make holes, similar to those produced by complement activation. The granzymes enter the target cell via the pores. This is that, here's the target cell. And it actually triggers that cell to self-destruct, apoptosis. And this is a scanning electron micrograph of a cytotoxic T cell actually killing a cancer cell. So they have photographic evidence that this is actually happening, that if there's a few cancer cells floating around in the body and your immune system is working correctly, at least your cytotoxic T cells, and they can identify the cancer cell before it reproduces, it can prevent the development of a fatal cancer. There's also these regulatory T cells or suppressor T cells. These dampen the immune response. They're very important to prevent autoimmunity, where the body begins to attack its own cells. And so this is actually a simplified summary of the primary immune response. I'm not going to go through this because we actually talked about it before. Now this is a discussion of organ or tissue transplants. There's four different ways, or four different types of tissue transplants. Autograft. This is the best because it's actually from your own cells. You will not reject that tissue because it's your own tissue. The next best would be an isograft. And so they can actually graft tissue from a genetically identical individual. Most of us don't have a genetically identical individual, but identical twins would have their twin that they could potentially have a graft from. Allograft, this would be from an individual of the same species, so another human being, either a living human being or a cadaver. You could take some tissue from a different individual to transplant. A xenograft, this would be from a different species, an individual of a different species, such as a pig heart, for example, which cannot be done because the tissue, the cells of the pig heart, a pig heart actually looks a lot like a human heart. But the cells are so foreign that the body would immediately identify it as foreign and reject it. They can, however, use valve tissue because it's just connective tissue. There's no cells. So they can actually transplant some tissue as long as it's not cells from a different species with success. The success of a transplant depends on the similarity of the tissues. Autografts and isografts are ideal donor tissues, and they're almost always successful. There's a lot of research going on xenografts. Okay, and they want to see if they can develop tissue that does not have these foreign markers on it, so it would allow the body to accept that graft. And so this would be like with pig hearts or something, where they can actually genetically develop a pig in order to have tissue that can be transplanted. the most common that we have would be an allograft. That would be transplant of a tissue from another individual of the same species, another human being. When this is done, when there's thinking about, say, a kidney donor of another person to donate a kidney, and this can be done, the donor has one kidney left and they're perfectly healthy. They can have a perfectly healthy life with one healthy kidney. So if they can donate their kidney to someone whose kidneys are failing, that would be just an amazing gift from one person to another. The only thing is they have to make sure that the ABO, the blood types, type A, type B, type O, blood types match other blood antigens, and the major histocompatibility complex proteins have to be matched to be extremely similar. It's not going to be identical, but you want it as close as possible. After surgery, they do everything they can to prevent rejection. And that means that the individual has to be on immunosuppressive therapy. And often these drugs can have a lot of bad side effects. The most common being corticosteroid drugs. These are steroidal anti-inflammatory drugs that suppress the immune system so that that person will not reject the graft. There's also some drugs that are anti-proliferative drugs, and these are often like really strong anti-cancer drugs, and they can also have pretty bad side effects, like methotrexate is one of them that can be used that can help an individual that's beginning to reject a graft or even some type of autoimmune disease. Immunosuppressant drugs. So there's also drugs that are not steroids that are suppress the immune system quite dramatically. Obviously, these can have very severe side effects. One of them being that if you develop an infection, you will not have as strong of an immune system and you can actually die from that infection. Or if there's a different kind of a cancer that begins to develop. When we reduce the immune system, we have seen that cancer can develop much more quickly. So when you're taking these immunosuppressive drugs or treatments, your immune system can't protect the infections. Again, they lead to death. Under the best circumstances, rejection will occur within 10 years of about half of the patients. There are some types of immune deficiencies, congenital or acquired. So these are when you have a very impaired immune system function. Actually, this is the congenital immunodeficiency. The most common one is severe combined immunodeficiency syndrome. This is SCID, and it's a genetic defect. And this is when that individual has a severe deficit in B and T cells. They have a defective adenosine deaminase enzyme that produces metabolites lethal to T cells. This is fatal if untreated, and the treatment would be a bone marrow transplant. Hodgkin's disease. Hodgkin's disease is actually a cancer of the B cells. It is considered an acquired immunodeficiency because a person is not born with it. But what this disease does, it depresses the lymph node cells. There's also acquired immune deficiency syndrome, abbreviated AIDS, otherwise known as AIDS. This cripples the immune system by destroying those helper T cells, the CD4 cells. The person experiences severe weight loss, night sweats, and swollen lymph nodes. And generally, the cause of death would be some type of opportunistic infection or a rare cancer. AIDS is transmitted by the human immunodeficiency virus, or HIV. This is transmitted via blood, semen, or vaginal secretions. This virus multiplies in the lymph nodes. It's often asymptomatic for about 10 years. When symptoms occur, that's when the immune system has collapsed. This can invade the virus can invade the brain, leading to dementia. The HIV, the virus-coated complex, actually attaches to CD4 receptors. It can enter that cell, the CD4, the helper T cell, it produces, or via reverse transcriptase. This is actually an RNA virus, so it can make then a DNA copy, called a provirus, to direct the host cell to make viral RNA, and that will allow that virus to reproduce. Autoimmune diseases. This is another disorder of the immune system where it's kind of like a hyperimmune system. The immune system is attacking its own cells. The treatment, again, would be suppressing the immune system. They may have received anti-inflammatory drugs, very strong anti-inflammatory. You may have heard of NSAIDs, non-steroidal anti-inflammatory drugs, such as ibuprofen, or which is Motrin, etc. You also have naproxen, Celebrex. There's all kinds of non-steroidal anti-inflammatory drugs. They have been discovering more and more that there are some pretty severe side effects when someone's taking those long term. They also can give them drugs to block cytokine action. And now the most recent development has been with the development of monoclonal antibodies. Humira, adalimumab, Remicade, infliximab. These are a couple of examples. I'm not going to go to the Wikipedia, but you might want to look at this yourself. I'm just afraid we won't be able to get back to the PowerPoint. But there are dozens and dozens and dozens of monoclonal antibodies. Anything that ends in MAB is a monoclonal antibody. These are considered, because they're produced by living cells, they actually can keep their patent. And so often, like if a company develops one of these drugs, it will not become generic because it's a biologic. That's what they call these, biologics, because it's produced by living cells. Therefore, they're enormously expensive for treatment, but they can save people's lives. They even use this for treatment of things like psoriasis. It's used for Crohn's disease and colitis, things like that. And also there are some that are developed to fight cancers. So there's many types of monoclonal antibodies. Many of them are used to treat autoimmune diseases. Another disorder of the immune system would be hypersensitivities. This is an immune response to a perceived threat that can lead to tissue damage. There's different types distinguished by the time of the course. When antibodies are produced with the immediate subacute hypersensitivities. T cells lead to what's called a delay hypersensitivity. And so that's when the body encounters something that normally is not going to cause a disease, but once the body becomes exposed to it, in response to some type of substance, the body will begin to produce antibodies. That causes an immediate hypersensitivity. And then if the body is activated to produce a T cell response, that's called a delayed hypersensitivity. Immediate hypersensitivity. This would be acute or type 1 hypersensitivities. They begin seconds after contact. This is when the interleukin 4 stimulates B cells to produce lots and lots of IgE, that immune globulin. The IgE binds to mast cells and basophils, and this leads to a flood of histamine. Histamine causes a lot of vasodilation of the small blood vessels, the arterioles, and a lot of capillary leakiness or increase in permeability. Each encounter with the same allergen can produce a more and more severe inflammatory response. This, for example, would be like if you are stung by a bee. You step on a honeybee and you get an allergic response. And it's a pretty strong response where you get, say you step on a bee and you're in your bare feet and your whole foot swells up. That's a pretty strong response. If you're stung again and your body is injected with the same toxin from the bee sting, it may be even stronger and the whole leg swells up. If that person is exposed again, it could lead to a fatal anaphylactic response. This is actually also occurs with killer bees, so that the type of toxin in that bee venom is no different from other honeybees. It's just that there's a huge, overwhelming amount of toxin injected into that individual at one time, because the bees are very they will swarm, they're very aggressive, and they will actually chase down a person, and when they're stung with many, many bees at once, it just overcomes their system, and they get a severe inflammatory response or anaphylactic shock, and that's how someone can die. So allergic reactions may be local or systemic. They're supposed to have an IC. Histamine causes blood vessels to dilate and become leaky. You also get a huge increase in mucus production, if you think of like in the airways, it can block the airways. Hives would be areas where the skin gets very red because of increased arteriolar dilation. The person will get watery eyes. Asthma is a type of allergy that is in the respiratory tract. So it's when the allergen is inhaled. If somebody has asthma, that's usually a chronic condition, so that they're particularly sensitive in their airway to certain allergens or conditions. A systemic response means throughout the body. This is called anaphylactic shock. Blood pressure plummets, and this is life-threatening. And actually, with either one, status asthmaticus would be a complete shutdown of the airways, where the airways they constrict and get filled with mucus, and the individual is no longer able to move air, and that's considered a medical emergency. Or with anaphylactic shock, this would be throughout the body, where the blood vessels just dilate, blood pressure drops to such a low level that they no longer can have circulation of the cardiovascular system. Anaphylactic shock, again, it's systemic. They also will get, if a person's in anaphylactic shock, they will also get constriction of the bronchioles. This is what happens with asthma. I may have said the blood vessels, but actually the little airways with asthma, these are called bronchioles. With everybody, they're bronchioles, but they're the smallest airways, and they constrict. They get very tiny and limit the amount of airflow. So this happens with asthma as well as during anaphylactic shock. With anaphylactic shock, vasodilation can cause fluid loss from the bloodstream out into the tissues, leading to circulatory collapse and then death. How do you treat it? The person must be given life support and the medication epinephrine. Epinephrine is a vasoconstrictor, so it will actually cause the blood vessels to become smaller, so therefore blood pressure will go back up again, and it can prevent them from going into circulatory collapse. And this is a very tiny figure right here, but it shows you the adaptive defenses in humoral immunity. Actually, how humoral immunity reacts with some kind of allergic response. Immediate hypersensitivity. The antigen invades the body. The plasma cells produce large amounts of class IgE antibodies against the allergen. The IgE antibodies attach to mast cells in the body tissues and any kind of circulating basophils. And these actually will release lots of histamine. And so that can cause a lot of vasodilation and an allergic response. This is showing you a subsequent or secondary response. You get more of the same antigen, invades the body. The antigen combines with IgE, attached to mast cells and basophils, and it triggers degranulation and release of histamine and other chemicals. The histamine causes the blood vessels to dilate and become leaky, promoting edema or increased fluid in the tissues. It stimulates the secretion of large amount of mucuses that will block the airways, and it can cause smooth muscle to contract. If the respiratory system is the site of the antigen entry, this is now called asthma, where these increase in mucus, the bronchial constriction, can limit the amount of air passage, and the person can asphyxiate and die. That's called status asthmaticus. Delayed hypersensitivities, this would be subacute, slow onset, 1 to 3 days, and it depends on the helper T cells. This is when cytokine-activated macrophages and cytotoxic T cells are causing damage to the body. Examples would be contact dermatitis, poison ivy. Also, the TB skin test. Many of you who may have had a job in the healthcare system, it's required that you take the TB skin test. In Europe, they actually have a vaccine against tuberculosis. So anybody who's had that vaccine will turn up positive. The reason they don't do that in the United States, give the vaccine that is they want to know for sure whether somebody's actually been exposed to tuberculosis, because it can hide in the lungs, in what they call little tubercles, and so it doesn't necessarily manifest itself for many years. But if you have this TB skin test and you have not had the vaccine and you have a positive test, it shows that you have tuberculosis. Developmental aspects. With age, as with many other things in the body, the immune system begins to wane, and so therefore we see more and more cancers develop as people get older, and also they're more fragile when it comes to fighting infection. And that is the end. Have a wonderful day, and I will see you with the next lecture or in class.